Okay, folks, let's do the first chapter PowerPoint. Let's go over this together. Um, the first thing it says is exploring life and science, the characteristics of life. So we're going to be talking about things like everything that's alive that we know of, it grows. Everything that's alive needs to take in energy to survive. They try to maintain homeostasis. That's a fancy way of saying maintaining internal conditions. Everything we know that's alive can reproduce. Um, One that's probably not in the book, everything that we know that's alive uses DNA. We don't know of any organisms that are not based on DNA. You might say, but viruses. Yeah, viruses are in that gray area. A lot of people don't actually consider them to be alive. But we'll talk more about that. Anyway, so we're going to talk about the characteristics of life, levels of organization. We're going to do things like, oh, well, you go from an atom to a molecule to a tissue to an organ system to an organism, that kind of thing. And yes, we're going to talk about the scary E word, but let's move on. All right, so levels of organization. Oh, they're actually starting with levels of organization. I would have thought based on the bullets, we'd do it the other way. Let's move on. All right, so levels of organization. Atoms are joined into molecules. Molecules are joined into cells. Cells are the smallest functional unit of an organism. Some things are single celled. I'm talking like protists, bacteria, things like that. We are multicellular. We have many, many cells. A tissue is a group of similar cells that perform the same function. I'm going to add here usually similar cells that are attached, that are adjacent to each other most of the time, meaning next to each other. And usually we have a basement membrane they're all attached to, but we'll move on. All right, so levels of organization. We're going from atoms to molecules to cells to tissues to organs to organ systems to organisms. That's what I had said we'd probably do, but they actually go past that, and that's nice. So after we have organisms, then we have species. The most common definition of a species, it's not the only one, there are many, but the most common would be the Ernst Mayr definition, the biological species concept. And that says that all the organisms that are similar enough to each other to reproduce with one another, they can produce kids, and those kids are not sterile, that is a species. Anyway, so we have species, and groups of different species together are a population. The populations, when we talk about all the things where they are, we get a community, and then an ecosystem will be all the living and the non-living things with it, and all the different ecosystems together form a biosphere. Okay, so levels of organization, definitely pay attention to this chart. You'll see stuff from it again. All right, so we're about to go through that table, that chart. An organ is made out of several different types of tissues. For instance, I've got a friend who's working with a really cool team at Shands. They are trying to grow new kidneys that could be used for organ transplant. And they've got 20 different types of cells that she's helping them look for. Make sure each type of tissue is growing back the right way so the kidney can work. Anyway, an organ system is a group of organs that work together. A good example of that would be your digestive system, where you don't just have a stomach and an intestine, but you have the stomach and the intestine and the, say, the teeth that help chew the food up, the esophagus that carries the food to the intestines, the liver that makes the bile that helps break down the fat, emulsify the fat, If you break it down into more drops, you have or surface area, makes it easier for the enzymes to work on. The pancreas is making those enzymes. All those things together are the organ system. They're working together. Each organ has a slightly different job, but they're all working together for a common purpose. An organism, it says a collection of organ systems. What I mean is you're not just a digestive system. You also have a circulatory system that carries the nutrients that you got around through the body. You've got a muscular system that moves you around. You have a respiratory system that provides you with oxygen. All the different systems together are what make you an organism. A species, a group of interbreeding organisms. That's a good definition. A population, the members of one species in a particular area, yes. Now, an ecosystem, all the community of populations interacting with the physical environment. What I mean by that is an ecosystem isn't just the stuff that's alive. So it's not like just the trees. It's the dirt the trees grow in and the water the trees use to grow and the water that the animals that eat the trees need to drink. All those things, the living and the non-living together. That's what makes an ecosystem. Anyway, and then the biosphere. Bio means life, sphere means round, circle-shaped thing. It's talking about all the life on Earth. All right, let's move on. So energy, the capacity to do work, is a basic physics definition. Most of the work that's happening in humans is pumping, is moving things around across membranes or through the body, or moving the body itself. And that requires, and that requires energy. So we get our energy, we are what we call a heterotroph. We get our energy by eating, okay? Hetero means not the same as yourself, and troph is what you eat. So heterotrophs are things that eat other organisms to survive. That's you, me, that's a lot of things. There are organisms that are called autotrophs, things that harvest energy from their environment and then make their own foods. Auto means you did it yourself. Troph means to eat. That'd be things like plants, like algae, stuff like that. But if you have to eat other organisms to survive, you are a heterotroph, and that's us. All right. The food we eat gives us nutrients that are used for building blocks and energy. We're talking about we eat proteins and we use the amino acids to build our own proteins. So let's move on. Metabolism is all the reactions that are happening in the cells. And where it says the ultimate source of life on Earth is the sun, that's because all of the energy, I should say most of the energy, obviously weird things happen in the deep ocean, but photosynthesis is what the autotrophs are using, the plants, the algae, some of the bacteria. They're harvesting light energy and turning into chemical energy, you know that. And they're the base of the food chain. They're what all the heterotrophs depend on. So then you have a primary heterotroph, that would be like the rabbit that eats the grass. Or you would have a secondary heterotroph, that would be like the fox that eats the rabbit that eats the grass. But at the bottom of the food chain, you got the grass. You know, you have to have the autotrophs at the base. Anyway, moving on. Homeostasis is keeping an internal environment the same. And it says most organ systems strive to maintain homeostasis. Great example of that. If your body is overheating, your circulatory system is going to send blood to the surface of your body. It's going to send it to the capillary beds in the skin, and that will let you radiate heat away. Even more, you're going to sweat on that skin if you're too hot. And as that sweat evaporates, it'll cool the skin, which cools the blood. And then you can deliver much cooler blood back to the body core, help bring the temperature down. Great. So that's how we deal with heat. What do we do when we're cold? Our muscles shiver to generate heat. It's almost like it's the muscle version of rubbing two things together. Your muscles are just moving, shivering to generate heat. Why? To raise the body core, raise the temperature of the body core. So homeostasis stasis means same condition. Homeo is we're doing it ourselves. We are trying to keep an internal body environment, and that could be the temperature, the pH, like your blood pH is between 7.35 and 7.45. It shouldn't go above that, shouldn't go below that. We try to keep everything level at a very steady state. And if it's not, if we go up a little, we'll react and bring it down. And sometimes we overshoot and then we have to bring it back up. That kind of situation where we're trying to keep things here and we go up a little, down a little, up a little, down a little, but they're all around this flat line. That's what we call a negative feedback system. Like, oh, this is bad. We can't let it keep going. Let's negate it. Let's bring it back down. Oh, that's too far. Okay, bring it back. Bring it back. When you wave around that line, that's the hallmark. That's the thing that tells you, oh yeah, you got a negative feedback loop. There are positive feedback loops. Giving birth would be one of them. You get a little bit of oxytocin. It causes uterine contractions, and that causes the release of more oxytocin, which, oh, here, let's do that again with the hand so it's easier. You have a little bit of oxytocin. It causes uterine contractions, which causes more oxytocin, which causes more uterine contractions, which causes more oxytocin, which causes stronger contractions. That is a positive feedback loop where things get stronger, stronger, stronger. Negative feedback loops are way more common in your body. The ones where we're trying to keep things at this steady state, and we might waver around a little above, a little below, but we're trying to keep things right there. That's what you see when you're trying to maintain homeostasis. Now, I do want to point out there are other animals that maintain homeostasis differently. Let's go back to the body temperature example. Remember, everything is homeostasis. It's not just body temperature. I gave you the example already of blood pH. The amount of oxygen in the body would be another example. We're trying to keep things in a certain condition. Well, what if you're an alligator? How do you maintain internal body condition? If it gets too hot, you go sit in the shade or in the water. If you get too cold, you go sit in the sun. They're still trying to maintain an internal body condition at a certain level. They don't have the ability to do it metabolically like we do, but they're still trying to maintain an internal condition. That's why it's one of the characteristics of living things. Alligators may not be like us, but they are living things, and they are going to maintain internal body conditions as best they can. All right. Everything that's alive has the ability to respond to stimuli. That means the example they're using is you get your hand off the hot stove. But you could have external stimuli, internal stimuli. If your blood pH, we talked about that before, if your blood pH gets too acidic, you're going to start, without even thinking about it, your body's going to start making you breathe harder. You're going to breathe a little faster, a little bit deeper. Why? Because you're blowing off carbon dioxide, and that reduces the carbonic acid in your body, which means you're getting rid of one of the acids. If you're too acidic, get rid of one of those acids. You don't need it. All right, so everything that's alive that we know of can reproduce. That means they can produce produce another generation of organisms. This could be bacteria that divide in half, or it could be a more complex organism that makes a baby that has to grow up and become an adult. All right, so everything that we know of grows. You might say, but the bacteria, well, it does get a little bit bigger before it divides in half. So even little single-celled things count. But obviously, this is human biology, talking about humans. As we grow, we get more and more and more cells. So your leg bones, little kid growing up, the leg bones grow longer They produce more cells. Those cells produce more ground material, matrix material, and then we calcify it. But let's move on. Development. It says all the changes that occur from fertilization until death. So they're talking not just about embryology, but also the development of puberty. The point is, you are not, it says all the changes that occur. You have changes in childhood, adolescence, adulthood. And so all the living things we know of change in some way. In some organisms, it's really subtle. If we go back to that bacteria example, yeah, there's going to be a change as the newly formed daughter cells get ready to divide themselves. They're going to have to make more of all the stuff in the cytoplasm they're going to divide up. So they change too. But that change is real minor. It's not like the change we have as we go from being a toddler to an adult. And where it says also repair after injury. We do a lot of cell division, and a lot of this is to replace old or worn out cells. That's where I believe they're going with that. All right, DNA. Everything that we know that's alive uses DNA. It's deoxyribonucleic acid. Yes, I expect y'all to be able to spell it. It's important. It is the famous, you see a twisty ladder in all the pictures. So you have the two rungs going up, 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 up. Sorry, I'll put my fingers close here. Two rungs going up, but they're twisted around each other. That's DNA, okay? I want you to know that little sections of that chromosome, of that piece of DNA, that have specific instructions how to make a protein or how to control another gene that makes a protein. But a gene is a small sequence of that DNA that has instructions to do something. And usually those instructions are how to make a protein. That's how you control different traits. Now, changes in the DNA are mutations, okay? Changes in the DNA are mutations. There are some that are beneficial mutations, good ones, some that are neutral. Most of them we now actually believe are neutral, and some that are negative. Now, which one is, whether it's good or bad or neutral, that can also depend on the environment. Suppose you had a grizzly bear in Montana, and it got a mutation, and that mutation deactivated a pigment protein, deactivated an enzyme that makes a pigment protein in their fur, and now you've got a white bear. Well, it probably wouldn't be that useful in the lower 48, but up in Alaska, that same mutation would be really useful. So whether you say it's a positive or a negative depends on the environment. Grizzly bears and polar bears are so closely related, they can breed together and produce offspring. That's why I picked that example. We think that what I just described is how we got polar bears. And then, oh, well, that's positive. They can blend in with the snowy background. They're good at that. Well, at that time, that would have been a positive mutation. If there's less winter and less snow around, then being white is probably not a good adaptation. And in that case, we would say it's not a beneficial mutation, but it's the same mutation either way. The same change, you made pigment, now you and your offspring don't make pigment, that occurs. Whether it's beneficial or not, that's up to you. We can talk more about stuff later. If you want really wild examples, we'll talk about nerve blocks in mice, things like that. But moving on. All right, so evolution is a change in a population over time. fabulous definition. I definitely want you to know that. I'll say it another way. That's not the way the book's going to say it. Pokemon is wrong. Those animals are not evolving. The little Pokemon things, imaginary animals, they are I shouldn't say adapting. Those animals are, oh God, why can I not think of the word? A mutation is a change in a population. They metamorphosizing. That's what I was looking for, metamorphosizing. So if it's a change in one individual, like a caterpillar metamorphosizes into a butterfly, okay? If it is a population that changes, like, oh, well, this mutation showed up in the population and they were all brown bears, but because the white ones have an advantage, they're a little healthier and they can produce a few more offspring than the others. So over 200 years, 300 years, the white bears become more common in this population and the brown bears less common. That is evolution. A change in a population over time is evolution. A change in an individual is metamorphosis. All right, so natural selection says the process by which evolution occurs. Basically, when a new variation occurs, it allows individuals to capture more resources. They have more offspring. I'm going to add a caveat. Usually. What if that same mutation also made them sterile? It's not going to spread through the population because they got no kids. That population is not going to be selected for and kept. Sorry, that mutation is not going to be selected for and kept. But in general, if a new variation, a mutation, makes it easier for you to do your job, then you're more likely to be healthy, you're more likely to have more kids, and that's more likely to be passed on to the next generation. All right, hold on a sec. I'm going to pause this because I just had a annoying pop-up. Oh, never mind. I'll deal with it now. Dealt with it. All right, so adaptation is a change over time. That's what population is doing. We already talked about that. So what's the difference in adaptation and evolution? Adaptation is one trait changing, usually is what we talk about. When we say, oh, well, they're adapting, usually that means the same thing as they are evolving. But I would also say we probably use the term evolution over a longer period of time, but it's the same idea. All right. This is a check your progress. So I'm going to be quiet here for a second or two. You can pause the video, see if you can answer these questions. If you can't answer them, go back and watch it again. All right, let's move on. So humans are related to other animals. Yeah, no big deal. Summarize the place of humans in the overall classification of living organisms. What we're going to be talking about there is probably taxonomy. And taxonomy, the big thing to remember, there's an acronym. Did King Philip come over for ginger snaps? That's domain, kingdom, phylum, class, order, family, genus, species. Anyway, we'll get through that. Then we'll talk about culture, how that's important and different from biology, and the relationship between humans and the biosphere. All right. So all of life is divided into 3 domains. The 2 first ones listed there, the bacteria, the archaea, they're stuff that doesn't have a nucleus. All right. Collectively together we call them prokaryotes. Prokaryotes are single-celled organisms. They've got no nucleus. Eukaryote, you have to understand, karyo is the Greek word for kernel, like kernel of corn, and eu is true or with. So basically, we're divided into the bacteria, the really old stuff that looks like bacteria, and eukaryotes, the stuff with a nucleus. That's what the division means. So prokaryotes, they got no nucleus. Eukaryotes, they have a nucleus. Why did I mention that kernel of corn? Well, before we discovered that some cells had a nucleus and some didn't, we didn't have a word for nucleus. So they just stole the Greek word for kernel of corn. Because it's like, oh, it's this little thing and it must be important. So karyo. Anyway, but what you need to remember is eukaryotes are things with a nucleus. And what I'm saying is eukaryotes, there are below domain. We had domain, kingdom, phylum, class, order, family, genus, species. Bacteria, the domain bacteria has one kingdom. It's called bacteria. The domain archaea has one kingdom. It's called the kingdom archaea. The domain eukarya, which you're probably familiar with, you know, the things with the nucleus, it has a kingdom called plants, plantae, and a kingdom called animals, animalia, and a kingdom full of fungi, as we call it fungi, and a kingdom called protista, which is this huge grab bag of everything. And some scientists are now subdividing that up further. Like, oh, we shouldn't call an amoeba, because right now you have an amoeba and a 100-foot tall kelp are both considered to be protista, kingdom protista, part of eukarya. And a lot of people are correctly saying, it's pretty weird to have no, we need to create new kingdoms. But guess what? Not everybody agrees on that yet, and so you don't have to memorize extra kingdoms. Right now we have 3 domains: bacteria, archaea, eukarya, and each and the 3 domains we're dividing up into 6 kingdoms. Bacteria has kingdom bacteria. Archaea has kingdom archaea. Eukaryota has kingdom animalia, plantae, protista, and fungi. That's it for us. All right. Let's move on. Oh, and there's the pretty picture. I could have just gone to the pretty picture. All right. So understand that plantae is plants, and animalia is animals, and fungi is fungi. And protista is a grab bag of everything from microbes to really big stuff. But all of these things have a nucleus in their cells. Almost all their cells. Like, there are a few exceptions. Like you and I, our red blood cells don't have a nucleus for reasons we'll talk about later. But yeah. Okay, so let's move on. So the domain eukaryota, that's us, the stuff with a nucleus, is divided into plants, fungi, animals, and protists. Yeah, we did that. You know that. Okay, we have some pretty pictures to show you what they look like. Archaea and bacteria, you know, are both single-celled. You know, they both don't have a nucleus. There are a lot of things that when you do these are the parts of the cell, and you hear about parts of the cell. Those are usually eukaryotic parts. Bacterial cells are much simpler. They don't have mitochondria. They're about the size of a mitochondria. So they're small, they're simple. They don't have all the membrane-bound organelles that we talk about for the complex cells like we have. All right, so eukaryotes, that's us. We have plants, and okay, yeah, you know about plants, algae, moss, great, you've seen those. You know that they photosynthesize, so they are autotrophs. If you look right here, kingdom protista, this is the one they said was a weird grab bag. They're like, oh, slime molds and water molds and algae. Yeah, but you have to understand, excuse me. Excuse me. You have to understand that algae includes kelp that are 100 feet tall. So we've got big things here, not just pond scum. Okay. Which, by the way, would probably be classified, most pond scum, they used to call it blue-green algae, but they now call it cyanobacteria. So they actually moved those into bacteria. which is sort of weird to me because it shouldn't be hard. It's like, it's got no nucleus, it's got a nucleus. You should know which one's a bacteria from the get-go. If it's got no nucleus, it's over here. It's one of the prokaryotes. If your pond scum has a nucleus, it's a protist. Anyway, but let's move on. Then we have fungi and animals. That's pretty straightforward. You already know that we're talking about mostly multicellular stuff. You know, you have to ingest food to survive. Okay, so most organisms in Animalia are invertebrates. And they say earthworms, insects, mollusks. Yeah, insects. It's insects. The crustaceans are everywhere. And the land crustaceans would be insects. Obviously, the ones in the ocean, you're probably going to have more crustacea. Not probably, you're going to have more crustacea, you know, things like crabs and isopods and lobsters and such. Anyway, most animals are invertebrates. They don't have a nucleus. Sorry, they don't have a backbone. So an invertebrate is a thing without a backbone. You, me, we are vertebrates. And so we have a backbone, a vertebral column. And our nerves run through that. Our nerve cord, our spinal cord runs through the vertebral column, which protects it. All right, so if we, and I'll tell you right now, I said, did King Philip come over for ginger snaps, which is the domain, kingdom, phylum, class, order, family, genus, species, where it says vertebrates are fish, reptiles, amphibians, birds. Those are all in the phylum Chordata. So did King Philip, that's the phylum Chordata. The vertebrates are phylum Chordata. And come, did King Philip come over for ginger snaps? Come is the class. So we have class Mammalia, class Aves for birds, class Reptilia for the reptiles. So anyway, let's move on. The ones like us, the vertebrates have hair, fur, mammary glands. Those are mammals. Okay, class Mammalia. And all right, so humans, raccoons, seals. Interesting choice. All right, let's move on here. All right, so humans are most closely related to apes, the Pongidae apes. We're talking chimpanzees, like the bonobo. But we have highly developed brains. Our pelvis is slightly different, so we walk upright. Creative language skills, totally true. If you're wanting to talk about animal language stuff, I'm totally down with having that conversation. Email me or talk to me. Stop in when I'm on campus. I actually was an intern back in my college days in a language lab with people who were trying to imitate the Yerkes Primate Center studies that were done with Kanzi. They're trying to mirror those using dolphins. And so the little bit of time that I got to spend hanging around with dolphins was in a language lab. And so I'm happy to talk about animal language things. And the short version is not nearly as well developed as humans in terms of grammar syntax. Yes, I know about Lou Herman and the dolphin showed syntax. I know 3 people who worked in that lab. But we'll move on. Anyway, tool use. Yeah, so what we're getting here is humans, apes, common ancestor. The book is harping on that. I know that's super controversial for some people. I'm not meaning to pick fights, but I do want you to understand what the book is saying, what most scientists are saying, and whether you agree with it or not, you need to be able to understand the concept. Basically, we share Hmm. I'd have to look it up to be sure if it's 96 or 98% of our DNA with pygmy chimpanzees, but we're pretty close. Um, the DNA is really similar, and a lot of the physiology, the way our bodies develop and how they work, which is what we're talking about in human biology mostly, very, very similar. All right, moving on. Okay, culture is activities or items we pass on from one generation to the next. Animals have that, but it's not nearly as well developed as it is in humans. We're talking about the beliefs, the values, the skills, the arts and sciences they have. Um, anyway, uh, we'll move on. You don't need me to start quoting Neil Stevenson at you. It's slightly tangential. All right, so the biosphere. We're part of the biosphere. This is all the living things on Earth. We're talking about up in the sky, in the dirt, in the ocean, all the living things. And we are living things, so we're part of that. All right, I'm going to pause for a second, see if you can answer these. If not, stop the video, roll back, watch the last section again. Okay, let's move on. So now we're going to talk about the scientific method. Talk about control groups and experimental groups. Talk about peer-reviewed journals. Talk about the type of information you have in graphs. The ability to read graphs is super important. A lot of the mistakes we have in understanding science from people not reading graphs. Anyway, then we'll recognize statistics and statistical significance. All right, so science is how we learn about the natural world. It's not just like some people act like it's a belief system. It's not. It's a method. It's a methodology. Our goal is to be objective and base everything on fact. We're humans. We don't always reach our goals, but that is what we're trying to do. That's what true science is. So the process of using science is the scientific method. It goes through a series of steps. The first thing is you make an observation. This could be something that you see, something you can measure. Then you form a hypothesis, an explanation for what could be happening. Now, this is real important. I want you to know that most people, when they say, oh, well, that's just a theory, or oh, I have a theory, they don't. They have a hypothesis, a possible explanation of what could be happening, which is, it's an idea. That's a hypothesis. When scientists use the word theory, it's something they've tested the heck out of. It stood the test of time. It still explains everything. That's a theory. Very different than the average person, average public. Oh, well, that's just a theory. No, 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 that's a theory, dang it. That's something we believe. We've tested that. I'll give you a good example, one that's not controversial. Gravity is a theory, all right? We have a pretty good understanding of gravity. We treat it like, oh, it's a fact. Gravity is still a theory. Anyway, and I have great confidence that gravity will work. So a hypothesis is a guess, a possible explanation for what could be happening. So you make an observation and then you're like, oh, maybe that's because of this. There's your hypothesis. I'll give you an example. Oh, wow, look, there are a bunch of dead fish along the edge of the river. Oh, I bet the paper mill upstream is releasing poisonous chemicals into the water. That's just an idea. Yeah, it's a hypothesis, a possible explanation. If you don't do water tests, if you don't find the chemical, you got no proof of anything. Now, inductive reasoning is when someone takes the facts or the observations and they, oh, well, that could be true because of this. Or in the example I had, oh, there are a bunch of dead fish. Oh, we know that factories release chemicals. Maybe they're releasing dioxins and that's harming fish. That would be inductive reasoning. You're combining facts that are known with some ideas. Our goal in science is to make a prediction that we can test. So you're not doing science if you're not making something that is testable. And when I say testable, I mean a prediction that could be proven false. Has to be falsifiable. In other words, oh, I bet that factory is killing the fish. Well, you need to be able to test and see, because it's possible you test and they may be releasing chemicals, but they're not the chemicals that are harming fish. So if I was going to test that hypothesis, I'd run two kinds of tests. If I was going to test the hypothesis that the chemicals from the factory were harming the fish, I'd do two things. One, I would test to see what chemicals are in the water. And two, I would then in the lab see what level of exposure to those chemicals might be harmful to fish in the lab. Because it's also a case where it's also a possibility where, oh, well, they're releasing chemicals, but they're at such low levels they're not causing any harm. This is why you make the hypothesis, oh, the chemicals from the factory are harming the fish, but that hypothesis means nothing without a test. A hypothesis has to be testable, it has to be falsifiable, and where it says it makes a prediction, an if-then prediction. So if the chemicals from the factory are harming fish, then I should be able in the lab using the same chemicals to get similar problems with the fish. All right. So inductive reasoning is when you're adding a bunch of things into the pot, taking this fact and this fact and this fact, putting them all together, adding them into the pot to get the stew of ideas. They'll give you a prediction. Deductive reasoning, that's where the if-then comes in. Deductive is, well, if this is true, then that should also be true. It says experiments are designed with experimental variables and responding dependent variables. Dependent variables are the results. And it's easy to remember that because the results depend on what happens in the experiment. So experimental variables would be in the case of my fish story, the level of chemical we expose the fish to. You might have a control group which has no exposure to that chemical. Their water is fine. And then you might have a group that gets a small amount and a group that gets a bigger amount, a group that gets a really heavy dose. And you could, hold on a second. I'm going to get a cough drop. There we go. You could have a model organism. They're using the example of mice. And yeah, those are used in animal experiments, and we are talking about human biology, so that'd be a good example. Oh, wait. I am going to pause this right now. My battery's running low. So. Okay, I'm back. We were just talking about experimental variables versus dependent variables. You know that the results depend on what happens in the experiment. That should help you remember that dependent variables are a fancy name for the results. Experimental variables are ones that we're going to manipulate in the experiment. Test groups are groups that are exposed to differences in the environmental variable. Control groups are not. So if in the example I was using before, fish, an exposure to a chemical, we'd have one group that has no chemicals in their tank, and then we'd have other groups that had increasing amounts of those chemicals I was wanting to test in the tank. Why do you have a control group? Well, in this case, it's a really good example. It makes sure that what you think you're testing for and what you think your results are showing really is what you're showing. If you have fish die in the groups that have chemicals, then you might be tempted to say, oh, well, see, that chemical, just like I thought, it was poisonous to fish. But if you don't have a control group, then you don't know if something else is killing the fish. Maybe you have bad oxygen saturation in the water, not enough oxygen. Maybe your water's not clean enough. Maybe you're just bad at keeping fish. So if you don't have the control group, you can't say, oh, well, all those deaths are due to the thing I was testing. If you have a control group and all the animals are fine, you can say, oh, well, look, it's not a problem with my technique for keeping fish, it's definitely the chemical. On the other hand, if in the control group you then had deaths of fish, you would know that there's something else going on. Anyway, and a model organism is an organism that could be used in an experiment. A lot of times we use model organisms because if we're dealing with human biology, we want to be ethical. There are things that we can't run on humans. There are other concerns too. Model organisms are used whenever it's really expensive to run an experiment. Anyway, let's move on. There we go. All right, so we get results from the experiments. You already know that the results are the dependent variable. Results in the forms of data can be shown in lots of ways. Graphs are a really popular method, line graphs, bar graphs. I want you to understand that the understanding how to read them carefully is a way to avoid being manipulated by people who are using graphs. All right, so here we have a graph, and the graph shows over time variations in blood cholesterol levels. And you can see cholesterol levels on the y-axis. You can see time on the x-axis. That's pretty straightforward. Let's go. So statistics are how we interpret data. Statistical data includes standard error or standard deviation. That shows you how uncertain we are of the value. Let's go back to the previous one, the previous slide. When you look right here at this data point, you might say, oh, that's what the cholesterol was. When you look at this data point, you might say, oh, that's what the cholesterol level was. In reality, it's possible that the cholesterol level could be as high as this or as low as this. And that's the deviation or standard error. Those error bars tell us what the confidence interval is. You could think of it kind of like in political polling when they talk about the margin of error. We say that candidate X is ahead of candidate Y, but that difference is within the margin of error. In the same way, we're saying that this is lower and this is higher, but in reality, if we had the highest possible thing that we're confident it could be here and the lowest possible here, there's no difference between the two. Now, so in other words, this one, this one, this one, we think are different, but we can't say for certain. But this and this, what's happening in week 4 and what's happening in week 2, there's no overlap at all. There is a difference. So the standard error bars tell you that you probably wouldn't want to talk about the difference between say week 2 and week 3, that's very little difference. It could be that there's nothing at all, but the difference a week later is dramatic and profound, and that you could write up. Anyway, statistical data shows us how certain or uncertain we are of a value. The bigger that bar, the less confidence we have. And statistical significance is used to, it says here, determined to evaluate the probability that some results are due to chance or some other experimental variable, and results can be published in a scientific publication. Yeah. We usually won't publish unless we can be certain 95, I didn't mean to touch that. If we could be 95% certain that there's a real difference going on, then we would publish. All right, so scientists look at the data in order to make conclusions about a hypothesis, and that can help us make another hypothesis for another experiment after that. It's a circular process. At the conclusion you make, you can use it to start further research. All right, so a theory, I talked about this before. A theory is an accepted explanation that, and the book doesn't say it's here, but it really should, that has lots of experimental evidence behind it. It's not just an explanation of what's happening. It's an explanation that has evidence, all right? If we say something is a law, that means that it is very, very accepted. Now, that can still be true if people don't like it. The book is talking about the law or the principle of evolution accepted by the overwhelming majority of scientists. Totally true. The vast majority of scientists accept this. To me, talking about this is a law or this is a theory, that's splitting hairs, because like I said, gravity is still treated as a theory. Gravity is considered to be a theory, but nobody doubts it. So according to the way that they're laying this out in the book, they would say, oh, well, that makes it a law. I'd rather just leave it as a theory. Anyway, but moving on. So let's go through some of these in a controlled example. A hypothesis, remember, is a explanation, usually unsupported, not always, but you have an observation, you're explaining it. So antibiotic B might be better than the current antibiotic at killing microbes. That's a hypothesis. Now, the control group, it says subjects with ulcers who get a placebo. That means a pill with no medication. A placebo is something that resembles the experimental variable, in this case, whether you are getting a dose of the new thing. And then the placebo resembles it, but it is not that. So in a controlled study, the one they're using, antibiotic B better than antibiotic A, that's the hypothesis. They have a control group that gets no medication. They get a little pill that does nothing. And then they have two experimental groups, each of which gets an antibiotic. One gets the old one, antibiotic A. One gets the new one, antibiotic B. So they have three groups. And they're talking about a double-blind study. A double-blind study is really important. A blinded study would be one where the patients don't know if they're getting the good one or the bad one. That's important because they might change their behavior if they are not getting what they think is the treatment they would like. A double-blind is one where neither the doctors nor the, I should say, neither the medical professionals nor the patients who are the study organism, either one knows what's going on. The PIs, the principal investigators, the guys who designed the experiment, they know, but they haven't told the doctors. That way there's no bias in how the data is collected. The doctors aren't like, oh, no, I'm thinking that if you look at this, there's either one or two right here. Oh, they have the new improved. Oh yeah, that'll only be one group of bacteria. I mean, I know it's kind of lumpy shaped, and there's no judgment call like that if the people collecting the data don't know. So that's what the purpose of a double-blind study is. And a conclusion would be what you come to after the experiment. And I think this is oversimplified because it might take multiple experiments to get a decent conclusion. Anyway, let's move on. Now, okay, the book has their what was just described on the previous slide. They have they now have a picture of it showing it, and in this, see if you can answer the questions. If you can, we'll continue on. If not, pause the video, go back, rewatch. All right, let's move on. We are going to talk about challenges that face science. One would be telling the difference in technology and science. Technology is the application of science. And then talk about limitations of science, challenges of science. All right, so technology is the application of science to human interests. Biodiversity is the number and relative types of species we have, how much variation there is in their genes, and how many different ecosystems where they live. Extinction is when a species dies out. And they're right, we're not happy about how quickly this is happening. We appear to be in a 6th mass extinction right now. Oh wow, there's a cone snail. There's a scary creature. That's a snail that has highly toxic venom. One of the toxins in the venom is a special fast-acting insulin that can throw the throw prey into a low insulin, sorry, a low sugar coma, basically a insulin-induced coma. It's very effective. And there are some people talking about trying to find a way to refine it and use the dose, use it as a fast-acting insulin for humans. That's a separate thing. Moving on. All right, emerging diseases. So this was written before the pandemic started with the coronavirus. And it's talking about H1N1 swine flu, H5N1 bird flu, SARS, MERS. These are all recent things. When I say recent, I mean like since I was in college that we've had these breakouts. SARS was when I was in grad school, and that was started from it appears to have come from eating bushmeat from palm civets in China, but it was a similar, SARS was a severe acute respiratory symptoms virus, or sorry, syndrome virus. It was a very similar, everyone having to walk around, wear masks. It killed a lot of people. It started in Asia, and it did get into the US. It was pretty bad in Canada for a while. Anyway, so we have new diseases that are coming back, then we have ones that are coming back all the time. We keep getting outbreaks of Ebola. That's a re-emerging disease, the one the book mentions. That actually appears to happen from people eating bushmeat that is often bat, giant fruit bats. And I know it's easy to, oh, well, that's gross, and da da da, like with civets or with bats. Or What we can say is that there would be a more accurate and enlightened way to say this is people got to eat, and you'll eat what's available if that's all you've got. But if you can avoid eating wild animals, you probably will have fewer disease outbreaks because there are standards for how you have to keep domesticated animals, and there aren't standards for wild animals. Anyway, just an opinion. So new diseases or reemerging diseases can come from increased exposure to animals. Globalization makes them spread faster. What that means is we can now hop on a plane and travel anywhere in the world. A good example of that would be with the current spread of COVID. If you may recall, early in this year, though I'm filming this in 2020, earlier in this year, we cut off the spread. Our government tried to cut off the spread of COVID by limiting who could come in from Asia, but then the disease came in from Europe. The limits they put on Asia didn't stop the disease because people could easily hop on a plane and fly to Europe and spread it there, and then hop on a plane to the U.S. And then infected people hop on a plane and fly to the U.S. because everything is globally interconnected. Because people can move so quickly from one place to another, diseases can move very quickly too. And it says some things come when disease vectors mutate and change hosts. That is exactly how we appear to have gotten COVID. That's exactly how we appear to have gotten COVID-19. There are lots of coronaviruses. I hate when people act like the only coronavirus is the one that's causing the current pandemic. But the fact of the matter is, it's closely related to coronaviruses that have been found in wild animals, pangolins and bats. And we think it probably mutated. Now, there's some people who are sort of conspiratorial who think this was done by humans in a lab, but it doesn't matter either way. A very small change took place in the DNA, which made it better at infecting humans than it used to be. And that caused all the problems. Regardless of where or how that change came, I don't want to fight with anybody about that. I'm not an epidemiologist. All right, so let's talk about climate change. Climate change can alter normal cycles of climate due to human activity. If we're getting more carbon in the air, carbon dioxide in the air, it is increasing temperature. You might have noticed that we had more hurricanes this year than last year. We have more fires this year than last year. The climate is changing. I don't know how people would deny that nowadays. The climate is changing. And so things are getting warmer. I really don't like the term global warming, though, because that implies we'll always make things warmer. In some places, it's not going to make the world warmer. It's going to make it drier. Unfortunately, that'd be places in the Midwest where we grow a lot of corn and wheat and things we use to feed the country. In other places, it's going to make it possibly wetter, which could do things like increase the number of mosquitoes, and then you increase exposure to disease from the mosquitoes. That it's not just global warming, the temperature's changing. A better term, it's more syllables to say, but anthropogenic climate change would be a better way to word it. Anyway, so we have a scientific consensus. Most scientists believe that the climate is changing, and in fact, I'll be more clear than that. 97% believe that the climate is changing and warming is happening. But let's move on. Okay, so this is again a check your progress. I'm going to pause. You see if you can answer these, at least know what the book says. I know some of the stuff that we've been talking about is controversial and you may not like it, but you need to at least be able to understand what the book is saying and why it is saying it. But I'll pause, you can check, and then we'll continue. All right, let's move on. Oh, and that is the end. Okay, that is the end of chapter 1. Excellent. Okay.